All right, so this is going to be the end of it. We are going to be done after this. I won't cover any more material, and I've already written the test to end here. Okay? We're covering 30. No, I won't cover 31 or 32. However, they will be for bonus points that you can do, and they won't be on the exams. Yeah, I'll turn it into a bonus assignment. Yeah. And you'll get the bonus points for it. Because I'm definitely not making it to 31, am I? Definitely not. So 30, protein biosynthesis. So translation, you may remember this, is the process of protein biosynthesis. It's named because the 4-letter nucleic acid alphabet is then translated into the 20-letter protein alphabet. The basics of your protein biosynthesis are going to be the same across all linked kingdoms of life that have been discovered on this planet. Your mRNA is going to be decoded in 5' to 3' direction, 1 codon at a time, and the protein is going to be synthesized in the amino to carboxyl direction. The amino acids will be attached to specific tRNAs. Remember the T's, you can think of that as being Tay. I actually think that would have been a much more elegant naming, but I didn't get to make that decision, I guess. Not old enough. So this right here, the polypeptides, are going to be synthesized again by successive additions of the amino acids to the carboxyl end, the carboxyl terminus. And so here we have a tRNA that's attached to our first amino acid. We join it to the next one right here. And again, that is at the amino side that gets joined. And then we get another one, and this is going to join up at the blue end again, right here, which again, this is going to join at the amino side. Everybody see that okay? Fantastic. So, your biosynthesis for the long proteins is going to require a low error frequency because you're going to do this thousands of times for a single protein oftentimes, and you're going to do that millions and millions of times per day, at least. Some of these you'll do it millions of times per week, but it depends on the amino acids and the proteins. So the translations, these must be accurate, yet they have to be fast enough to meet the cell's needs. The E. coli translation can take place at a rate of 20 amino acids per second. Anybody ever heard the enemy of getting anything done is perfection? Yeah. So keep that in mind with all of this. If you're like 1 in 1,000 times, it's going to take longer than 1 in 100 time error, right? And if you have 1 in 10,000 times, it'll be exponentially longer than 100 time error. Okay? So the error frequency of this epsilon is going to be the 10 to the negative 2. This is intolerable even for small proteins because there's probably going to be an error even in a small protein, right? 10 to the negative 3, this usually leads to an error-free synthesis of an average size protein. But when you get to a large protein, you're almost assured to get an error in it. And so 10 to the negative 4, this would be allowing for accurate synthesis of large proteins, but you'd still be very rapid. However, keep in mind that 10 to the negative 4 error rate, this still means you're probably seeing an error every few large proteins, right? Yeah. So that's actually a pretty high error rate as in terms of like, if you were to have an error rate like this for products delivered on Amazon, you'd actually have some problems, wouldn't you? Yeah. So anyways, the accuracy of the protein synthesis, again, this is going to be that accuracy. This would be your number of amino acid residues. This is your probabilities here in terms of error-free protein. And so right here we have 10 to the negative 4. And when we get up to the 1,000 amino acid residues, we can see that we have like a 90% chance of getting an error-free protein. So the majority of the proteins that come off of this are going to be error-free, but what does a 90% chance also mean? It's like a 1 in 10 has errors in it, right? Yeah. So you have a pretty decent number of your proteins in your body that are going to have errors in them. Now, are all errors problematic? No. There's quite a bit of room on the proteins that it really doesn't matter if you have an error, right? And so, yeah. The codon, this is going to be your 3 coding bases on your mRNA template. The fidelity of your protein biosynthesis is going to require the accurate recognition of these codons. The transfer RNA, or tRNA, this functions as the adapter molecules between a codon and an amino acid. You can think of them a bit like the tags that you would put on something, especially if you ever have one of those automated sorting systems. You guys ever seen one of those? No? Okay. You never seen the Amazon automated ordering? Aw. Okay. Man, I'm just having one of those days where every single example I come off. Nope, never seen those stock calls. All right, so the anticodon, this is the portion of the tRNA that base pairs with the codon. And again, it's just going to be the anti-sequence that is going to hydrogen bond to that codon sequence. Remember, these will be your bases, not the amino acids. So each tRNA is going to be a single chain. This will contain between 73 and 93 ribonucleotides. Each of the tRNA sequences can be arranged in a cloverleaf pattern in which 50% of the nucleotides are going to be base paired. And this only looks cloverleaf if you do this in a 2-dimensional structure, by the way. Though I guess it does look like a really fucked up cloverleaf if you look in 3-dimensional. Okay? So think of a really messed up looking clover, okay? So that 3-dimensional structure, this is going to be L-shaped. The tRNAs, these are going to contain unusual bases. They'll have the methylated or demethylated derivatives of A, U, C, and G. The methylation will impact your hydrophobic character, actually. And so right here, we actually have the methylated version right here. And this one is the dihydrogen one for the uridine right there. Okay? So methylated or demethylated versions. So the 5 groups of the bases are not going to be base paired, but they'll participate in the hydrogen bonding interactions. So we'll have the 3' CCA sequence right here. Everybody see that okay? Gray one. Fantastic. By the way, can you guys see that better now? Big improvement or small improvement? Moderate or small or big improvement? Big improvement? Okay. I'm trying to figure out if I need to come in here before classes every time and lower the blinds. Because we don't have bright enough projectors to overcome that. So this next one is going to be the T-Psi-C loop. That's this right here. Named after this group right here. Everybody see that okay? And then there's an extra arm. And so we have the extra arm on here. That's this one right here. This is actually variable. It doesn't have a specific length it has to be, though theoretically there would be a minimum length that it can be. Okay? The DHU loop. And so that's this one right here. And then there's the anticodon loop. And this is the anticodon loop here. Which one has the anticodon, by the way? Hopefully that's pretty obvious. The anticodon loop, right? Yeah, this one down here. Yeah. Okay, so it has an L shape if you look at it from the side while it's in 3-dimensional space. Can everybody see the nice L shape look here? Okay, and remember that this was this right here. Okay. Up here. This was this part right here. Right there. Okay. Now, this again is the anti-codon loop. That's the one that's going to match with your codon. That's the tag part. That's the part that's going to get read in the machinery which we are calling the ribosome, right? Now, this portion right here is the portion that's going to end up getting connected to your amino acids. That's your CAA terminus. That's going to be over here. So again, these are the different portions in the three-dimensional structure. Again, that's that CA terminus. This is your anticodon loop. Please note the colors are actually matching. So that yellow here, that's down here. Everybody see it okay? Okay. And then you're going to have your loop there. That's the DHU loop right there. This is the green right there. Again, that's the red that led up to that CCA terminus. Your extra arm is right here. And again, this doesn't really matter that much how large that is. It just needs to be large enough to get down here. Okay? Because it's not particularly meaningful space. Notice it's not hydrogen bonded to anything. It's just a loop, right? This part right here, again, is that hydrogen bound portion that'll lead up to your anticodon loop. So the 5' end of the tRNA, this is going to be phosphorylated. The 5' amino terminal residue is going to usually be the PG. What does the PG stand for, by the way? So G's G, right? What do you think the P stands for? Phosphorylated, yeah. When we had been D, that would have been the deoxy, right? Yeah. So the activated amino acid is going to be attached to the hydroxyl group of the adenosine in the CCA region of the acceptor stem. The anticodon loop is going to be the loop near the center of your sequence. And so right here we actually see the ester linkages. They're going to couple our amino acids to your tRNA. Again, this is your CCA arm of your tRNA right here. And this is the amino acid being coupled to it. Okay? And in this case, we have the amino acyl tRNA because they are coupled together. Anybody see that R right there? Which amino acid is it? All of them. Yeah. When we see an R like that, that means, yeah, we can be anything we want. Okay. And so the anticodons are going to base pair with the codons. Some of the tRNA molecules can be recognized by more than one codon. And so again, you have a degree of non-specificity that is going to reinforce your mRNA specificity. Does that make sense? I.e., you'll have several matches that it can be because those are places that you have a tendency to have errors. Okay? You don't quite have as tight of a fit. So the wobble and the wobble hypothesis. Redundancy or degeneracy of the genetic code indicates that the recognition of your third base of a codon is sometimes less discriminant than the other two. This is known as the wobble. So the wobble is the steric freedom in the pairing of the third base of the codon. The wobble hypothesis is going to be the established hypothesis that predicts the binding of anticodons to codons. And so this right here would be the first base. These are the allowed pairings at the third bases over here. So the first base of the anticodon, the third base can only be G. The first base here, A, this can only be U. But when we get to U, we can actually fit A or G. Ooh, you weren't expecting that, were you? Good stuff, huh? Okay. And so that's your wobble, right? We already have some wobble there, don't we? Yeah, a little bit of mismatch. This G, this is going to fit U or C, and then I can fit U, C, or A, any of those. This is actually the inosine-cytidine base pairing right here. And then we can actually see the inosine-uridine base pairing right here. And notice that it can still match. It's not that far apart, is it? Yeah. It's a little off, but not that much. When you're talking about three things coming together, it's not that surprising, is it? Okay? And then we get the inosine-adenosine base pair right here. You've probably all experienced wobble, where you have like the screw together mops and stuff, and they don't quite connect. There's a little wobble to it, right? In the middle. Yeah. It's the exact same thing. Okay. It's a little bit of air, a little bit of wobble room. So the codons that differ in either of their first two bases, these must be recognized by different tRNAs. Part of the degeneracy of genetic code is going to arise from your imprecise, or wobble, in the pairing of your third base of the codon with the first base of the anticodon. So the first base of your anticodon determines the degree of your wobble. The ribosomes, these are going to be the huge RNA protein complexes that will consist of those two subunits, which will assemble around your mRNA, right? We've discussed those a bit before. One subunit is going to have 3 universally conserved bases in its RNA molecule. These are going to form hydrogen bonds only with the correctly formed base pairs in the codon-anticodon duplex. And so we can actually sort of see this right here. Here we have the anticodon base. Here we have the codon base. And you can see here, if it matches up, what happens is this brings this right here, this oxygen here, into close proximity. See that? And we can start matching up our hydrogen bonding between this group right here, which is the 16S. This is that conserved adenine right there. And so that's actually the mechanism that's going to enforce that matching. Okay, that's a large part of that mechanism. I know. We had an extra hydrogen bond we didn't list, did we? Okay. Okay. Anybody have any questions about that? What would happen if we get the wrong one in here? Yeah, so the wrong anticodon comes in, and it no longer perfectly matches, or matches well enough. Yeah, this would either be closer or further, right? And then this hydrogen bond wouldn't be as stable, and so it'll come out easier, and you'll have a higher likelihood of having the correct one attached for longer. Okay. And so your amino acids is going to be required for protein biosynthesis. First, though, they must be attached to the specific tRNA molecules. Man, nobody has ever seen a like a factory operation where they have hoppers, or a like the automated robots that stock. You guys never seen that? Anybody ever stocked anything for like a store? Yeah, only you. Okay. I mean, it's the same thing, right? They have little tags and they have a little machine that reads it. Yeah, you go ding, and it tells you where to go. There you go. Okay, it's basically how robots work too. They put tags on the things. If they screw up the tags, guess what? It goes in the wrong place, right? Yeah, and it gets really messed up. So the attachment of a given amino acid to a particular tRNA, this is going to establish your genetic code. The formation of the peptide bond between the free amino acids is not thermodynamically favorable. This will take energy. So the amino acids are going to be activated by forming an ester linkage. This will occur between the carboxyl group of the amino acid and either the 2' or the 3' hydroxyl group of the terminal adenosine of the tRNA. So this is going to form that amino acyl tRNA. That's the charged tRNA. That's the one we had looked at in the figure before, okay, with the R group. So again, this is your amino acyl tRNA synthetase. Oh my God. My talking has given out. It's done for the semester. Wow. Anyways, synthetase, this is going to catalyze your activation of your amino acids. Does the name make sense to everybody? It's the complex you're synthesizing, right? And that's the activated form of the amino acid, or amino acyl now, because you've bound it to this. The first step in the formation of the amino acyl adenylate, or amino acyl AMP. So we have the amino acid here. This is going to react with ATP. This is going to give you your amino acyl AMP. Now notice this will provide energy, right? Because we're cleaving off those two phosphate groups. Remember how this was not thermodynamically favorable? Guess what? We're getting thermodynamically favorable, aren't we? Okay. Okay, so the amino acyl group of your amino acyl AMP, this is going to be transferred to a particular tRNA. This is going to form the amino acyl tRNA. Again, that tRNA will have the correct anticodon that matches that amino acid specifically. But for now, we're not looking at specifics. We're looking at the overall. And so this will give you the amino acyl tRNA. This would then free that AMP. So this is the sum of that activation and transfer steps. Remember, again, we're coupling this ATP to provide that thermodynamic drive, that thermodynamic energy. So the reaction is going to be driven by hydrolysis of the phosphate, just like almost all the reactions, if you look far enough. One ATP equivalent is going to be consumed to form the ester linkage of the amino acyl tRNA. One ATP equivalent is actually consumed to drive the reaction forward. The amino acyl AMP intermediate does not dissociate from the synthetase. This means that the same amino acyl tRNA synthetase catalyzes both these steps of the reaction, and so it was the same enzyme for both parts. Okay? This doesn't actually come off in between. Otherwise, you'd have a very highly energetic reactant that could, transfer its phosphate to a lot of different things, right? Keep that in mind. That would break apart pretty easy. The acyl adenylate intermediates are right here. So this is the acyl adenylate intermediate right here. This is the amino acyl tRNA, and this is the fatty acyl-CoA right here. So the acceptor of the acyl group, this is going to be CoA and fatty acid activation. The acceptor of the acyl group is going to be tRNA and amino acid activation. The energetics of both biosyntheses are very similar. In fact, you can make a lot of parallels between all of the pathways between these, right? Sort of set it up with a phosphate, have this catalyst, usually the same enzyme, that then is going to assemble it into the correct layout, right? And then use that to link onto something, build something. Okay? So the energetics, again, are very similar between the two. Both are made irreversible through this hydrolysis of your phosphate, or the pyrophosphate, the PP, right up here, later on. So each of the amino acyl tRNA synthetase is going to be specific for each amino acid. These will be the actual things that recognize and join these things together so it's correctly tagged. To avoid coupling to the incorrect amino acid, the 3-anyl tRNA synthetase is going to contain a zinc ion at the active site. This actually binds the amino and the hydroxyl group of your threonine. The val, similar in overall structure to threonine, but lacks the hydroxyl group, so it's not going to be joined to the tRNA that is the specific one that would have threonine. The SER is going to be occasionally linked to the tRNA that would be useful for threonine, but because of the presence of the hydroxyl group, it's going to do that. This is actually looking at these groups, so you can see. Notice again, the valine has a very similar structure. See that? In terms of groups. But we don't have a hydroxyl group. On the other hand, the serine actually has a not that similar structure, right? But it has the presence of the functional group, the hydroxyl group. And so you can get mismatches and linkages that would happen where instead of a threonine, you get a serine. This right here is actually the functional group and how it matches up. Again, this is that zinc ion. Notice it's bonding right here. And this is going to, again, be what selects the amino acids for the synthetase right here. Notice that these right here are the different amine groups attached to this zinc. Right here we have the oxygen right there. Okay? Everybody see that okay? So the 3-anenyl tRNA synthetase, this is going to have an editing site that's going to hydrolyze. You can actually hydrolyze your serine tRNA. This will improve the fidelity to less than 1 mistake and 10 to the 4th. Because the 3-enene contains an extra methyl group, it's actually sterically excluded from the editing site. And this is how you fix that mistake that happens occasionally. Okay? And again, we're taking advantage of that stereochemistry, that difference between these two, where this can't fit in the same sites this would, right? Okay. So the amino acylated CCA arm, this can swing out of the activation site. It can then enter the editing site to remove the serine. And so what happens is when you're bound, what happens to all molecules all the time? They're constantly removing, retaching, moving around, right? Diffusing. And so if you're bound to a serine, it's going to be able to move up into that editing site, right? Whereas the 3-enene has that methyl group, it won't be able to fit into the editing site, and so it just keeps bouncing off and never gets into the editing site. And so between that editing site and the activation site, we actually get pretty good fidelity. For biological systems, quite good fidelity. And this is this right here. This is the active site. And then this is the editing site right here. You can see that this can swing up in here if it's a serine. And this is a abstracted, so you can actually see it a little easier. Okay? And again, that would be that CCA arm. Can everybody see the tRNA right here? The L structure? Yeah? Okay. Okay. And with the methyl group, it won't actually be able to enter the editing site, and so it'll just stay in the activation site until it releases. Over here, if we don't have the methyl group, it can swing up. So the kinetic proofreading, this is also going to increase the fidelity of your protein biosynthesis. Kinetic proofreading is going to be the mechanism that's going to incorporate the irreversible step to facilitate removal of incorrect substances. This will locks in the equilibrium, favoring the correct product. The selectivity is going to remove incorrect products to increase specificity further. Kinetic proofreading is actually used by the amino-acyl tRNA synthetase. It can be used by ribosomes. It can be used by additional molecular processes. For example, we have the DNA replication that uses this as well. And so this is the intermediate amino acyl adenylate. Here, if we look at the blue versus the red, that's going to be an important part. And so we have the enzyme plus the preferred substrate. And what we can see is over here, we get a lower energy intermediate. See that? Okay, and then right here, this activation energy, this goes lower here, and then we go to this intermediate here. This would be lower here as well, right? Okay. Now, this right here would be alternative substrate for the red. Okay. Now, notice a little bit of an interesting thing here. Which one's faster here? So here, there's not really much of a difference, is there? Does everybody remember what kinetics and speed depends on? It's the difference between your reactant here and the height of this part right here, which is that transition state, right? The higher the transition state energy, the slower it goes, right? Lower the transition state energy, the faster it goes, right? Okay. So if we look here, what we can actually see here is that if we look at that alternative substrate, that thing actually goes faster, doesn't it? See that? What do you notice about the blue one instead? Yeah. And so this right here is going to have your wasteful products, which will be the free amino acids and AMP, because this is going to be the slow step in this reaction in the first place. And then this right here for intermediates can actually go back and forth a lot faster. Okay? And so, so your side reaction is actually faster than this correct product. And so you're going to end up getting these wasteful products out much faster. Here we'll get the correct product stuck here. Okay? So, okay. So was it clear that this is actually what you want right here when I'm talking about this? Maybe we should approach this from a different angle. Okay, so right here would be where you start, okay? And the blue, again, is going to be the preferred substrate, the one we should be binding. Over here, this would be the alternative substrate, the red, i.e. the air, okay? Okay, so we basically match up for these two. There's not that much difference between them. See that? Okay. And so when we look at this right here, what we're doing is the correct product has the lowest energy for this, and it can have a further processing step, right? For example, we had the editing step up here. Remember that? So this would be like an editing step here. Okay? And so because the energy barrier for the incorrect product is lower, it can go into the editing step much faster. It doesn't have as much time to release and be the wrong thing. And this editing step is going to free those back up into the waste, but your waste is going to be free amino acids and AMP. Now note that this is wasteful. What happened to the, where did the AMP come from originally? It came from ATP, right? So you are wasting energy every time you're doing this. But what that does keep you from getting is this intermediate right here, which is the incorrect one. Okay? So what you're using is that kinetic rate for a second editing reaction, a second splitting reaction, it coming back apart. You're using that kinetic rate and making it so the correct product is the one that comes out the fastest. Okay? Is that making sense? Does everybody understand that this is the actual thing we're looking for here? Okay. Did I lose anybody there? No? Fantastic. So the amino acid tRNA synthetases, these are going to be the true translators of your genetic code. They're going to assign a particular amino acid to a specific tRNA. The synthetases are going to recognize the tRNA partners. This is primarily on the basis of their anticodons. These synthetases, these may also recognize other aspects of tRNA structure that vary among different tRNAs. Many of the recognition sites are loops that are going to be rich in unusual bases. Okay, a little survey real quick. So in the previous classes you had where you talked about translation and transcriptions, what did they usually say was the part that actually did the translation? Did they usually say it was like the ribosome that did that? Yeah. Yeah. So the part that actually does the translation and turns it into the language of amino acids into bases is actually these synthetases. There's nothing else that recognizes that match of any real note. There's a little bit of error correction later, but that's basically it. Okay? So once it's tagged, it's going in as the amino acid. Okay? And that anti-codon-codon matching, that's already been done for the translation for the amino acid, right? Because that's still a base. Okay. So the synthetases, these might also recognize as other aspects of tRNA structures, vary among different tRNAs. I've already done that. The CCA arm is going to extend into your zinc coding containing activation site. And so you're right here, we can actually see, again, this is the editing site, this is the activation site. This right here is that CCA arm. That actually extends all the way in and is actually what flips up and down here. Okay? And that's actually one of the reasons why it's important that it's only single strand. Do you ever wonder why it's only single strand? For a few bases there? No? Is your brain going, Dr. Clausen, I'm done? Just okay. Yeah, so if it had been double strand, it could not have any flexibility to move, right? It would have been too rigid. So that's actually why that needs to be single strand, in case you're ever wondering. So the enzyme is going to interact extensively with the acceptor stem of the tRNA and the anti-codon loop. And right here's the anti-codon loop. Notice it also matches up with this synthetase, right? See that? And so you're reading the anti-codon loop down here. This is then matching the correct amino acid up here to your CCA arm. And typically there's an editing site that if it has the wrong structure and it can move to the editing site, it's going to get eliminated. Why do you do that? Why is that? What are the 3 levels that we're using to check this and actually attach these things? What are the 3 identifying factors? Okay, so if I went like, this person has brown eyes, long hair, and is 5 foot tall. What are the 3 factors I'm using to identify them? Yeah, hair, eyes, height, right? Okay, so there's 3 factors in here too that are being used to identify them, right? There's, is it bigger than the activation site and can it fit in, right? That's one factor. The next factor is the fact that does it hydrogen bond in such a way that it can make the full hydrogen bond set, right? If it can't make the whole full hydrogen bond set, it either is not going to attach or it's going to end up in that editing site. Now, what happens if I get something too small? Like in the case of our serine. Fit the hydrogen bonding, it can then go in here, it's small enough that it can go in here. I'd be screwed, wouldn't I? If I stop there. If it's too small, that's not going to get excluded. If it has the right hydrogen bonding, it's good to go, isn't it? So the last thing I need is the things that are too small and can still do the correct hydrogen bonding here. I need something that will take those things that if they're small enough, they can move up into this and then get detached. Fix that error, okay? These other 2 are the easiest, fastest ways to do this. This last one is sort of a desperate last measure, okay? Theoretically, this actually exists in a number of other sites that we've talked about with like the N-oxide pockets and that sort of stuff. And that is that they have to be big enough to be able to reach this other binding site, right? That was actually similar in purpose. Now, again, these are going to have the tRNAs. They're actually going to have multiple recognition sites for amino acid tRNA synthetases. It's actually not just the anti-codon loop that has a recognition site. So these yellow portions are the recognition sites that are going to be recognized right here. It's a lot more recognition than you thought, right? And so that's going to determine the recognition of the synthetase and then give you the correct amino acid linked. So class 1 and class 2 synthetases, each of these are going to include enzymes that are specific for 10 amino acids. They're going to bind different faces of your tRNA molecule. They're They're going to have a CCA arm of a tRNA in different conformations, the hairpin for class 1 enzymes, and helical for class 2 enzymes. The acylate different groups of the terminal adenosine of your tRNA, the 2' hydroxyl for the class 1 enzymes, 3' hydroxyl for the class 2 enzymes. This has an exception for the class 2 though, which is the tRNA for Phe, PHE phenyl. The enzyme. Everybody's comfortable with this, right, by the way? Yeah, this is the amino acid that tRNA should attach to. Okay, so we bind the ATP in different conformations. These are going to have different numbers of monomers, 1 for class 1 enzymes and 2 for class 2 enzymes. And so this is actually your classification and subunit structure. This will be for the amino acyl tRNA synthetase. This one is for E. coli. And we can see these right here. These are class 1s, these are class 2s. Okay? Again, this is the class 1, class 2. You can see that they bind in slightly different parts of the tRNA. And so right here we have the class 1, and this is the tRNA without it. This is the CCA arm that's going to have that amino acid attached, right? And over here we see that it binds on this portion right here. Everybody see that okay? Okay. Does this currently look like an L to you? No. Why it does not look like an L? It's perspective, right? So it's coming out a bit towards you. See that? Yeah. If we wanted to see the L structure, we'd have to rotate it a bit. Okay? Okay. And so this is the other one. Note that, again, this is not a perfect lineup with this one. You can really tell from the loops. That's the easiest way to tell. Right here. They're a little bit different, but very similar. Okay? We're not quite lined up, but we're very close. A little off. And here we can see the class 2, and it's pretty obvious it attaches at different points, right? Because it's basically almost the mirror image, right, of the class 1. Okay. Anybody have any questions about that so far? Okay, so the ribosomes, these are going to coordinate the interplay of the amino acyl tRNAs, the mRNA, and the proteins. These are the ones that will actually be the machinery that takes your nice little tagged amino acids, fits them in with the mRNAs, and then connects the things and then it's out to states. So the E. coli ribosome is going to have a sedimentation coefficient of 70. By the way, that's what that 70S actually meant. And this is going to be composed of 2 subunits. We have the large subunit, which is 50S, and then the 30S smaller subunit. By the way, does that add up to 70? It does not. So the 50S subunit, so this is going to be composed of 34 proteins, 23S rRNA, and a 5S rRNA. Yes, those are the 23 and 5S rRNAs that we looked at in the previous chapters. Okay. The 30S subunit, this is going to contain 21 proteins and a molecule of 16S rRNA. And again, that's that 16S rRNA from the previous chapter. This is actually the different subunits. So right here we can see the 50S, right here we see the 30S. And then this is how they actually join together and create your 70S ribosome. Okay. By the way, if anybody's wondering, yes, this is X-ray crystallographic data of what these look like. They are somewhat abstracted to make them easier to look at, but that's usually true of all X-ray. Okay? I know. Don't you love that? Come on, you're going to miss it. Who else is going to make you look at all the proteins? This is how they fit together. You guys are totally going to miss this. You're going to go, I want to see proteins. And then you'll look them up yourself because it's not actually that hard. So the 2/3 of the mass of the ribosomes is actually RNA. The 3 RNAs that are incorporated into this are the 5S, 16S, and 23S. They're critical for your structure and function of your ribosome. They're formed by the cleavage of that primary 30S transcripts and then further processing. They're folded into defined structures. They're going to have short intramolecular duplex regions. The ribosomal RNA is going to be the actual catalyst for protein synthesis, with the ribosomal proteins making only a minor contribution. And it's actually widely thought, though I think they're still working on it, trying to get some sort of more evidence to this theory, that there was actually a version of ribosomal RNA that did this without proteins. That was a precursor before. So the ribosomal RNAs are going to form the intramolecular base pairs. This is actually what that looks like. Can you guys see those duplex regions right here? And how they're going to get bound together? Is that okay? And again, remember, structure is function, right? And so when we look at structure versus function, again, we have a 3-dimensional structure. It can then bind to things. And we can bring those into a reactive structure that will be a complex in the correct orientation, right? And so it sort of fulfills our baselines for enzymes as we have known them. So the 3 tRNAs are going binding sites in the ribosome will allow the formation of peptide bonds. The aminoacyl site is the A site, the peptidyl site, P site, the exit site, E site. You can't guess what the exit site's for. Exiting, yep. No, right? The aminoacyl site, what do we bring in there? The amino acids bound to what? RNA, yeah. And then this is the peptide or peptidyl site, right? Yep, you got peptide. Yeah. The mRNA fragment's going to be bound with the 30S subunit. That's that small one that's first going to attach. That one right there. Each of the tRNAs then get in contact with the 30S and 50S subunits. At the 30S end, the tRNAs at the A and P site are going to be bound to the mRNA through the anticodon-codon base pairs. So this right here is actually going to again be the 3 binding sites here. E, exit site, P, peptidyl site, A, aminoacyl, right? Okay. And notice, you can pretty much figure out what's going to happen here just from the naming, okay? We're going to have our tagged amino acids come in, right? They're going to end up being joined into peptides, i.e. a single-dimensional protein, right? And where do you think that protein joining tends to happen? Has to happen somewhere here, right? And then we're going to eject the stuff that we don't need anymore, right? And so, exit site. Yeah. I guess I'll stop here because I only have a minute left, and I'm starting to get into stuff that needs to get, like, cohesive. Yeah. Anybody have any questions, concerns, comments, party tricks? Party trick?
No, I don't have one.
Aww.
What chapters is going to be on this test?
Up to 30. Yeah, so if you look at the previous deal, whoa. So it's 20. No, 19 is not on tests at all. No, we didn't. 19 is just not, in it anymore. Yeah. 21, 22, 28, 29, no 23, no 24, dropped all that. Yeah. So it's only the ones I actually covered in class that'll be tested on. Okay? Everything else that I don't cover in class is bonus assignments. And I'll end up moving the 31 and 32 to bonus points. Okay? Yeah, we'll finish chapter 30. Nothing else than that, though. Martina, at some point I need to talk to you about your forms, because there's one you need to pick up and make sure you have. Oh, shit.